attendees are in listen-only mode.
I'm not going to do a lot of introduction today in order to kind of keep us on pace and on time, but feel free to take a look there to learn more about each presenter. And in addition, at the end of the session, depending on how much time is available, we'll do a Q&A with all four of the presenters. Okay, which one are we going to? The Michael Beach. Yep. Okay, good. All right. I'd like to introduce Michael Beach, who is the Associate Director for Health and Water at the Centers for Disease Control and Prevention. Thanks, Michael.
Okay, so am I walking with this?
There's your mic, yeah.
Okay, well, thank you very much. I had a great time getting here that it took me over 12 hours sitting in the Detroit airport at 2:30 in the morning. So I got here about an hour ago. But I really appreciate feeling so welcome when I see that you renamed the track out the front there with my name. So I do appreciate that. And I think, you know, I've always really gotten a great welcome from the Beach Conference, and it's really wonderful to come here and see that. So anyways, what does CDC like to do? They like to count things, and I think the reason for that is that it's the ability, if you count something, you can use that data for decision making. You can convince people to do something. If we don't count, if we don't look, if we push things under the carpet, that means we don't do anything. And by we, I'm saying policymakers. Policymakers act if you have data and other sorts of things behind it to do it. And so I think it's really important for us to be thinking about that in the future. So I want to talk about some of the kind of not data heavy here, but get you thinking about some of these things. We have all these other august individuals here who will be talking about some of this in more detail. But let's think about, if we think about ambient water, and I'm going to talk about kind of estuaries, marine, as well as fresh water and that, so you can think about what some of the issues are. CDC oversees the waterborne disease outbreak surveillance system, in which all of you at the state and local level kind of feed outbreak investigations and that back to CDC, so we can look at trends and that sort of thing over time. So back to the early 70s, we have data. When you look in the data, wow, there's virtually nothing from the Great Lakes, nothing from marine beaches. And so it's really a big gap for us. And why do we think some of that happens is that you have really huge beaches. And so when people come to the beach, what do they do afterwards? They come from actually an average of about 20 miles up to 100 miles away, and then they disperse. And so if there's any transmission of disease, we tend to lose it, and it doesn't get picked up as outbreaks. But we know from the EPA studies that of all the Great Lakes beaches studied and the marine beaches, what do you see? Have any of you looked at those? Sarah Collier's paper just came out, kind of summarizing this for all the beaches that were looked at. And what you see is there is always a basal level of diarrheal illness and ear infections and so on in those individuals who don't go in the water at all on the beach when you interview them. It's always increased above that baseline. We think it's baseline diarrheal illness. Above that, if you actually get in the water and put your head under the water. So every beach, when you follow prospectively, you can find illness there. But we never see this show up as outbreaks, or virtually never. I think there's one out of Door County in Wisconsin that we see. And so from a pathogen standpoint, HABs being a different issue. So just keep that in mind as we start to think about some other indicators that we might look at, especially from a climate change angle, because we're talking about water. We're talking about potential increases in water temperatures and nutrients and so on. And what can that kind of show us? So obviously, we know all of this. It's about increased or decreased water availability. Boy, we can walk both sides of the fence here, depending on who we're talking to. Extreme weather events, you know, droughts and floods, increased use of recreational water. When people get hot, where do they go? They go to the beach. And we know this from many instances during heavy heat spells and that. People, you don't want to close the beaches during those times because it really actually increases other social issues if you don't let people defuse at the beach. So increased water temperatures, and we have a lot of thermophilic organisms that we would like to think about. Can we track them in that? So let's talk about the Vibrios. And for those of you who don't know, just quickly, infection with a species from the family Vibrionaceae, a variety of different species. We're not really talking about Vibrio cholerae here. The cause of cholera, because we don't really see that in the U.S. very much. First recognized in the 70s, it became a nationally notifiable disease in 2007. And so there's a variance here that you can get if you eat them, meaning they're in your favorite friend who likes to concentrate all the pathogens, mollusks and other sorts of things. If you eat them, you can get gastroenteritis. Worst thing, as you can see in the picture up here, you get this in a wound, and you can be in really big trouble here. So a number of severe wound infections, and again, leading to septicemia. So when you go to the beach, whoever used to be told, oh, go in the ocean, it'll clean the wound out. Like the ocean is sterile. I mean, so anyways, there's a lot of wonderful things growing there in that mix. They like the marine kind of low salinity habitats, and it really, warm waters, low salinity is key from a Vibrio standpoint. Okay. I might have, okay.
Okay, sometimes. Try it now.
Oh, it's on here.
Well,
okay, I got it now.
It should work on that, but I'll stay here in case it doesn't.
So risk factors for this, as I mentioned, it's about eating mollusks. They're concentrating all these pathogens and so on. Undercooked shellfish. So if you like those sliders and kind of raw things, I used to work in the hepatitis group, and we had freezers full of those. So we know that many of you love to eat those. Wound exposure to sea, I'm sorry, not me when I looked at that many of them. So wound exposure to seawater, though, is a classic for us from a recreational standpoint. About 1,000 cases are reported each year, but the estimates are about 80,000 infections occur per year, 500 hospitalizations, and about 100 deaths. This is especially with Vibrio vulnificus. You get this as a wound infection leading to septicemia, and there's pretty high risk of death. It is not a good infection to get. So I'm going to
work. I'll just hit it.
Yeah, and so what we've seen over time here is increases. This is from the COVIS data that comes into CDC reporting Vibrio cases, and we've seen quite a dramatic increase over the past couple of decades. And so what I wanted you to just kind of think about is here, Vibrio parahemolyticus into Alaskan waters, 1,000 kilometers farther north than previously reported. In 2004, they started seeing outbreaks, so it moved up the west coast. It's also up the east coast, up to the Chesapeake Bay. Infections in northern Europe, which tended to coincide with these 4 years here, which actually, if you look at that, those were hot spells in the Baltic area during that time. So again, heat-loving organism. We tended to see peaks during that time in the first reports coming out of Scandinavia and so on of Vibrio infections. Hurricane Katrina, a lot of people were talking about cholera and all that sort of thing after Katrina. Well, you have to actually have cholera in the population for that. And so, you know, offshore and that sort of thing. So what people really saw here was you were wading in those floodwaters and everything. So there was a large increase in Vibrio wound infections. And then really, if you look at some of the great work out of Rita Caldwell's lab and others, these Vibrios tend to kind of track with El Nino events. So especially with cholera and other sorts of things, you tend to see during warm water changes that you'll see increases in transmission and outbreaks. Next one. So you've heard a lot about HABs. I'm not going to say very much here, except I do love these pictures. Who was at lunch and saw some of the pictures when the young ladies were in the green scum? This is the one I just got off the internet here, but I always thought people tended to avoid this. But clearly, there were some really great pictures of large crowds mucking around in this. From a marine habitat, you know, HAB standpoint, respiratory illness, foodborne illness, the National Outbreak Reporting System data from 2009 to 2013 shows most of these are foodborne. 58 foodborne outbreaks reported, 209 illnesses, 30 hospitalizations. Again, if we move to the freshwater side of things, as you've heard, you know, the cyanoHABs, really World Health Organization has provisional guidelines, 1 microgram per liter. And again, we know now that Toledo exceeded kind of not US EPA standards, but at least some global standards that were set when they had to shut off their water system for a few days. No federal guidelines for this. obviously a collection of different toxins. And the issue for us is this is a really tough one to confirm the illnesses. And so, what's the case definition? How do you confirm this and really link human illness to these events? And that's really what we're going to be looking at in the future. Go ahead. And so what we've seen is prior to 2009, 5 outbreaks were reported. And again, remember, this is outbreak, not event. It's outbreak, meaning 2 or more human illnesses. And so we really don't see that many in the waterborne disease system. Foodborne was what I was talking about earlier. 2010 to 2011, we saw more than we've ever seen before, 11 freshwater outbreaks in New York, Ohio, and Washington. And then the last 2 years, we see it drop back down to 1. And I think what we're going to see is this kind of, depending on how many you investigate and how it gets reported. Yeah, and so it's going to be, I think, jumpy for a little while, and Virginia Roberts will be talking next, I think, right, about we really want to bring back online the harmful algal bloom information system. It's got a new name now, but we need to collect these data. I think this is the only way to convince policymakers that we must keep moving. So Toledo, Ohio, as I said, first U.S. city reportedly exceeding WHO values, and there was a surveys done afterwards in that documenting illness, increasing in frequency, intensity, and duration, as you know, nutrient levels and everything doing that. And we are in the process of piloting this national surveillance system, which Virginia will talk about next. I just have to talk about amoebas. And I think this is an issue and I'm totally biased here, because actually the amoebas are in our group. But if we want an indicator of change, this is it. And so let me remind you what this is. Primary amoebic meningoencephalitis, brain infection, rare and serious brain infection. We've seen 138 cases, and we've been tracking since 1962 to 2015. And the way we can do that is most diagnostics in the country come through CDC because we have the capacity for it. Only 3 known survivors. 2 just a couple of years ago, the last one in 1978, so we're pleased with some change there with a new drug coming online. And this is something that, as you see in the picture here, it's about swimming in fresh water, warm fresh water. The amoeba goes up your nose. It's not a parasite. It's a free-living amoeba grazing. It's a cow down grazing on the microbial mat and on the bacteria, and then you get it up your nose. It crawls up the olfactory nerve and essentially then starts to live off of brain tissue. So it is called the brain-eating amoeba. Naegleria fowleri is its name. Most infections are lakes and rivers, and I'll talk about some of the changes there. But again, this is about going up your nose, not about drinking. So it's distributed globally. Wherever anybody seems to look in fairly warm water, you tend to find it. Luckily, we don't see as many infections. But clearly, this is a thermophilic organism. It's found in hot springs and other places, and so it's ideally adapted to basically proliferate in the warmer waters when other organisms don't do as well. So up to 115 degrees Fahrenheit, pretty warm water here. Next one. So what did we see here in 2010 to 2013? The issue for us is we've been tracking it for 50-some years. So starting in 2010, we saw the first case in Minnesota. This is 600 miles farther north than ever recorded before. Everything was in the southern tier states until that time. We've had a second case there, actually in the same lake. First 2 cases in Kansas, never seen it before. First case in Virginia since 1969. There were 2 lakes associated in Virginia. They had the perfect intervention. They filled the lakes with dirt. The lakes don't exist anymore. They saw no new infections from 69 until now. That's how you get rid of something, just get rid of the lakes. Not what we can all do. And then the first case in Indiana. So I hope you're feeling kind of what we do, which is it's coming your way from a Great Lakes standpoint. Next. And so not only, we haven't seen really increases in number of cases, what we see is the histogram kind of filling in. We see it moving north, and then we see changes in transmission routes. So 2011, what we saw is the first documentation in the country of people who uses a neti pot when they've got a sinus infection. Oh, good. I hope you know what kind of water to use. Yeah, so you know what kind of water to use, right? It's not about tap water. It's about distilled water. or boiled water that's cooled. So neti pot use, you know, it's a little teapot you put up, rinse out your sinuses. So these individuals actually self-inoculated with Naegleria. It had colonized, it moved into their water heater system, and it had colonized the residential plumbing system. The year later, we went to the Virgin Islands, and we saw, again, a Muslim male who had, in your Muslim male, many times you practice ritual ablution, where it's essentially you're putting water up your nose to cleanse prior to prayer. Self-inoculation again. The individual's home was colonized with Naegleria and essentially in the water that they were putting up their nose. Louisiana 2013, we had a child die in a slip and slide, you know, where you kind of jet along the water. Well, what's it do? It jets water up your nose. It's hooked to the hose, the tap outside the house. It's the first time that we've seen a treated water system supplying water that caused infections. This is harkening back to the Australian issues that were in the 70s and 80s when this was being transmitted. And then a couple of years later, the first death associated with this year, overland piping. So if you pipe water over land in the desert, it gets really hot. And what does it do? It selects for this type of an organism if you're piping it out of a river. And so in this case, they used the water for the residential plumbing system, fill up the pool, and one of the guests died from Naegleria infection. And we know the first, basically we've now in Louisiana, we have multiple water systems now because it was made, it was a governor's called alert, an emergency alert. All water systems in Louisiana are tested, and now we see a couple of water systems being colonized per year. We don't know about the rest of the country because nobody looks, but we know now that at least in the thing, yes, okay, next. Okay, and so in conclusion, just what I want to do, it's a very short presentation, but I hope what you're getting the feel for is there are changes coming. We can track these, we can start to use these data to convince policymakers and other folks that change is coming and we need to adapt for that. And so geographic ranges of pathogens are clearly changing, some indicators showing increasing numbers, changing transmission routes. I mean, this is, for Naegleria, this is since 2010 that all of this has occurred. So it's pretty dramatic change. Drinking water systems are now impacted. You know, I went to a community meeting in Louisiana. If you want to tell somebody a message that's really hard to take, it's the water's safe for drinking, but not to get up your nose. It could kill you if you do that. That's not a very acceptable message for a community. You essentially, so one death can undermine confidence in the entire water system. And so a very difficult issue to deal with. And that's what's coming in the future, I think, as we start to think about this more. But hopefully what you've gotten about this is it underscores the need to document, track, report, and publish these data to get it out there so that you can use them as decision makers on what to do. And I'm all about data supporting decision making. And we've got a really great group to acknowledge. And finally, if you don't like the CDC and what I have to say, you can ignore me. This is our official little note that we have to put on there. So thank you very much.
Thank you.
So I had dueling timers here. I'm not sure which time each group was on.
Right. So what we're gonna try to keep on track.
We have a few minutes.
While we're transitioning, you want to take a question?
We have time for one question.
One question. Yes. Oh, hi, Eric.
Yeah. Can you have them talk in here?
Sorry, the question was about tropical Cercaria, right? And what, I'm sorry, what are you asking about? You know, it's a great question, but we don't really track them. We know they're out there, but there's not a lot of tracking data, so we don't know. Good question, though. Anybody thinking about this Naegleria now? Hopefully, so. Okay, I think we're.
Thank you, Michael.
Thank you very much.
We'll have more questions.
Yeah, we'll have a little bit of time at the end. I'd like to introduce Virginia Roberts, who's an epidemiologist and the Great Lakes Project Coordinator at the Centers for Disease Control and Prevention. Thanks, Virginia.
Okie dokie. Well, I hear this is working again, so that's good. Let's see. There we go. So I think the first thing I want to do is I want to start by thanking some people. I don't have all of the names listed, but I do think that the work that I'm presenting on today is such a collaborative effort that we need to start by thanking some folks. So first of all, this project wouldn't be moving forward at the rate it's going without the involvement of the Great Lakes Restoration Initiative and its regional working group. There's also a working group of state and federal partners that I'll discuss later, which we call our HAB working group, and CDC surveillance partners who work on the surveillance systems, either from an IT development standpoint or from an epidemiologic standpoint at CDC. So Michael mentioned the Waterborne Disease and Outbreak Surveillance System. Just a little more background on that. It was initiated in 1971 with a focus on drinking water by CDC, the EPA, and an organization called the Council of State and Territorial Epidemiologists. Recreational water outbreaks were added in '78, and this really is that primary source of national data about the scope and effects of waterborne disease outbreaks among persons in the United States. The data from that system really go into reporting out summary reports, publications, data and statistics, information for health communications, the website Michael had up at the end, and developing programs, health promotion, and policies. So when we talk about harmful algal bloom or HAB-related exposures, typically they get classified when they come to us as being kind of in that waterborne realm, unless foodborne, of course. So how do we receive data on those outbreaks? It's through the National Outbreak Reporting System. This is an electronic reporting system that was launched in 2009. Prior to that, states sent us reports of outbreaks on paper-based forms. So this has really helped us with the exchange of information and communication with partners. It's voluntary reporting by local and state health departments, and the data are aggregated. So we're not talking about individual cases of illness. We're talking about, when there was an outbreak, you kind of lump all that data together into a single report. The surveillance is passive, so it's not about going out and actively finding an outbreak. It's about, when you find them, investigating and reporting them. And it's not an active response system. It's about reporting after the investigation is complete. Its focus is on human illness, so it's 2 or more human cases of illness linked by time, exposure, and illness characteristics. And that's what gets reported to the system. And in addition to waterborne disease outbreak data, we capture information on foodborne disease outbreaks and enteric diseases associated with other exposures, like norovirus and nursing homes and so on. So just to kind of emphasize, this is the general process of how data comes to us before it gets kind of summarized, published, and otherwise disseminated. So the key things I wanted to highlight here are, after people are exposed to some sort of pathogen or agent, if they get sick at some point in some way, the health department has to get notified, investigate the outbreak, decide that it is something that's reportable, enter it into NORS. So it really, this process really relies a lot on the state capacity to detect, investigate, and report outbreaks. And further, you see that really the health department is, our primary stakeholder when it comes to, or primary partner when it comes to actually that data coming into the system. And the reason I bring that up is when we talk about HABs, it's really, we're thinking about, and this is true for all water, but with HABs, we're really thinking about a different paradigm for the kinds of stakeholders we bring in. So what happens with HAB-related case surveillance in the United States? Well, some of you are familiar with HABIS, the Harmful Algal Bloom-Related Illness Surveillance System. This was a One Health system, looked at human illness and animal illnesses, and HABs as well. So that case data was reported to a system at CDC. But it only funded select states to improve HAB and HAB-related illness surveillance. And the activity is closed, so that data collection no longer happens. So we don't really have case data coming in to understand what's going on with harmful algal blooms. However, in FY 2013, the Great Lakes Restoration Initiative funded CDC to begin building public health capacity in the Great Lakes region, with a big driver being to collect better data to kind of inform Great Lakes restoration activities and assess kind of project impacts. One of the pieces of that is harmful algal bloom surveillance capacity. So one of our project activities is building a web-based reporting system for One Health HAB surveillance, essentially reviving HABIS and bringing it back on board. Like HABIS, So what we've been working on is the One Health Harmful Algal Bloom system, or OHABs. We had a little vote, and that one came onto the top. So it's a system, again, human and animal health data can be reported here. And one of the critical things about it when we talk about this One Health approach is that cooperation amongst, human health, animal health, and environmental health communities. This is kind of a novel system for CDC. We talk about One Health in other capacities, but we don't have a system that has kind of the animal and environmental health having such a stake in the game, I think, as we do with this. OHABS is looking at systematic collection of data on HABs and single cases of illness. It's informed by HABIS. It's event-based, so it's not routine monitoring of water. And again, it's a kind of after-the-fact state reporting. We're able to get the system online by tethering it to NORS, which is one of the reasons I talked about that earlier, and it's web-based reporting. So I guess the things, why are we doing this? Within that realm of informing GLRI and understanding HABs, I mean, we have a lot of questions that need to be answered to define, case definitions, understand where those exposures are happening, where, what kind of toxin concentrations impact illness, and so on and so forth. So I think we look to OHABs as a tool for understanding and helping us. And I think I say us as everybody in this room and beyond, kind of this idea of understanding and preventing HAB-related illnesses. I'll also point out that we recognize that this is an emerging public health issue with a lot of needs, including needs for clinical diagnostic testing, resources, increased awareness, and so on. But that's, those are opportunities. Once you've defined those, you can start tackling those. And I know there are a lot of people who have already started working on pieces of this. So this system, we can come together, and this can be another piece of moving the dial forward. So the timeline of OHABs has actually been pretty quick. We received our initial funding in fall of 2013. In January of 2014, we launched a working group of state and federal partners. By October of last year, we had started IT development. In May of this year, we brought on a full-time coordinator to facilitate our pilot and launch. And in August, we launched the system as a pilot for state users so they can start kind of interacting from this working group to see what they think about this system and give us input, and to also go out to federal partners and come to places like here and talk and get some input. So the working group, you can see it has state and federal partners. I've kind of shown all the state partners, and you can see that within our pilot system, or our pilot project, there's a lot of representation within the Great Lakes region, as one would hope, and we're really excited about that. Working group products include reporting forms, which then will lead to what we're used to program the system itself, and reporting criteria. So these are case definitions for suspected and probable and confirmed cases of illness, and also kind of an event. What kind of event would you report to the system if you were to? What are the bounds on that? Is it a certain toxin level, or is it, a visible bloom? How do you get to those things? And we expect this to evolve over time with more data. So now I'm going to show you a little bit about what the system looks like. I'm not going to do a full demonstration, but outside of today, I'm happy to do so, or to arrange something outside of this meeting itself, some sort of live demo. But the idea, when we talk about OHABs, we talk about reports, but reports actually have three different components. One is an environmental form, and one is a human case form, and one is an animal case form. So these are the places where you put information about illnesses and environmental data, and together that makes a single report. The process, in these next few screenshots you see, the system is tethered to NORS, so someone can log in through NORS, and so this is a system that states use to report again, so access is kind of controlled at that state level. If they do log in, here's my little pointer. So there's this little link on the landing page for NORS where you can then click and go to the system. Or there's a direct login option for OHABs, so that if you don't want to go that extra step, you could do that. Once you get to OHABs, there's a landing page, and then so you have a search section here, which allows you to search reports by an ID number, a state, a report date creation, or where the say a bloom occurred. You have a view and select reports section that has report IDs, Again, location, date created, author, and status, and you can sort this screen by those headers. You also have an option to delete the report, and you also have these 3 little nifty images. So this shows you that there's one kind of environmental form. In this case, there's one human illness form, and there's no animal forms kind of attached or reports into this report. And so you can see this is all test data as part of the pilot. We've been doing a lot of demonstrations. So we also have the area where you can create a new report. We're working on a data download feature because we know it's important to get the data back out to users and other people who need access to this type of information. A link back to NORS and a resources section. So if you wanted to create a new report, you'd click on the button to the right, and you'd have this pop up. So what you would do is you would enter a state report ID for a new report, and then you'd have the option to choose between either an environmental, starting with an environmental form, a human form, or an animal form. And if you were to choose one of these, the latter 2, you'd have to enter a case ID, otherwise that would be grayed out. And each state establishes how it's going to name its report, so it's easier for it to find those pieces of information later. Now the human form, so this is basically the general setup for every type of form. You have the title at the top, you have some quick links back to all of the reports and the report summary, which I'll show you in a minute. You have the case ID. You have the section here that kind of gives you some information on the case, the sex, the age, the author, the date created, and then some information about the environmental data for the larger report as a whole. So this is just a quick reference when you're in the report, and this sort of information gets populated as you fill things in. You have these headers along the top here, which are sections of the report, and you have then the subsections. So no personally identifiable data is collected in the system, and that's important because, well, basically it would be a non-starter for being linked to NORS if we couldn't because of the security issues tied there. But we didn't really feel that we needed to collect things at the level of, say, birth date or residence or phone numbers or those sorts of things. So you have a general section which describes the person and the dates of exposure. You have human exposure information. What were they doing? Where were they when this happened? What were their symptoms? What were the outcomes? Did they go to the hospital? Did they see a doctor? Those sorts of things. Clinical testing results. And we have in the supplemental info section, we also have a place where you can attach additional information if you have it that didn't fit within the report or you want to reference later. So that's the general design. Very few of these fields are required, mostly just a couple of dates and the location where the exposure occurred so that we can kind of keep track of these reports and manage them. Now, we said the form collects information about animal illness as well. The setup for the forms is similar. We ask slightly different questions about animals. The thing that I wanted to draw attention to is that we know that there are times when there are, you have a fish kill or you have a large flock of birds, and you're not going to have information on that you're not going to report every single animal. So there is an option to report a group of animals, and the questions are slightly different. And this was certainly a big part of the decision on this was based on working group feedback. Now in the environmental form, you have different sections. So you have the general section, which talks about where did the bloom occur. It has some linking variables that you could use to link to environmental systems, like hydrologic unit code, coordinates for where, say, a bloom occurred, that sort of thing. You have a bloom description, which you can put in information about health advisories or observational data about the bloom. Did it smell? What color was it? That sort of thing. Laboratory testing data about algal toxins. And you can put in information with links to other systems. And then you have the supplemental section. And I think really this is useful for things like pictures of the bloom, or if you do want to put in additional water monitoring data that you haven't entered in the form. Because as we said, this is not trying to replace existing systems for monitoring. So once you get through entering data, if you have a complete report, you have a section that looks like this, a report summary. that has the environmental information at top with some of the information that's been filled in, and the human and animal case information summarized here. If you want to add another case, you can go over here to the action section and do so. So this is just a really quick way to see and get at some of those subsections of your report. And now, just wrapping up, I really want to just kind of recap a couple of things, because we do get a lot of questions about when we show the system about OHABs. So it is event-based. We're not trying to recreate or supplement anything that's already out there. We're trying to build on that. It's looking at human and animal cases of illness and HAB events. Waterborne and foodborne HABs can be reported, but outbreaks will be reported in NORS. And just finally, just coming back to this idea of One Health surveillance, One Health surveillance really does require a community to make this work. It's going to have to go beyond the current paradigm for surveillance partnerships and reporting. Bringing in especially the animal health community, I think, is a huge piece of that. Capacity really extends beyond having a system. It's great to show you, that we have a system, but if there's nothing to feed into that and nothing to take the data and use it, then we don't necessarily have that capacity to address the issue of HABs. And we really look forward to moving this into kind of its full launch and building a system that can inform Great Lakes restoration efforts, mitigate health effects of HABs, and work, collaboratively to prevent illness. And that's it.
I think we probably have time for one question. Yep.
Are you going to be tracking any of the dermal effects of the HABs?
So
The question
here.
Go ahead.
Repeat it.
So will we track any of the dermal effects of the HABs? So if, for example, if there was a person who had exposure to a HAB or an animal and they had a rash, that could be reported in this system. Yeah.
This is right. Okay, Francy.
Great. And we will, again, have time for Q&A at the end. So now I would like to introduce Donna Francy, who's a hydrologist with the US Geological Survey, the Michigan Ohio Water Science Center. Donna?
Okay, thanks, Joe. Let's see if this works. So the beach monitoring community has been pretty successful at predicting E. coli concentrations based on predictive models at beaches throughout the Great Lakes. In Ohio, advisories are issued based on models at Lake Erie beaches through the Ohio Nowcast that's shown up here. So we were kind of wondering, if it works for Lake Erie, will it also work for cyanobacteria harmful algal blooms? We call them CHABs, cyanoHABs. There's different acronyms. And would it work, and what kind of information would it give us? Well, for one, it would help us to decide when and where to sample better for toxins associated with CHABs. And secondly, would it work like E. coli? Could we provide a CHAB real-time swimming advisory to the public? And then the third thing is not only recreational waters, but maybe we could help some of the drinking water plants in either adjusting their treatment or their intake options. So all of these things could potentially benefit from predictive models. So we initiated a study, and we sampled collected data at 8 sites, all recreational sites throughout Ohio, some Lake Erie sites, and some inland lake sites. And this was done over a 2-year period. And well, the first thing you want to do if you want to develop models, you have to identify if there are factors that are related to a human health concern. And of course, our human health concern here are the toxins produced by cyanobacteria, or the toxin concentrations. So that was our main objective of our study. But at the same time, when you start to develop the models, you find out a lot more about the systems. And you can use all these factors that you hopefully identify to understand what's triggering toxin production and the proliferation of the cyanobacteria. So we looked at a bunch of different factors, and these are some of the ones we looked at. We did analysis for cyanobacteria by molecular methods, qPCR, and also the traditional cell counts, where you look under the microscope. We looked at phycocyanin and chlorophyll pigment measurements, either handheld sensors that we brought to the beach or nearby continuous monitors. We looked at nutrient concentrations, and then we took into account the weather and hydrology. So we sampled in the area used for swimming, because that's what's important, obviously, the area where people are exposed to the toxins. We sampled throughout the recreational seasons in 2013 and 14. We sampled, the water depths were about 2.5 to 3 feet, so we sampled that top foot of water, and we composited it across the beach like you might do for E. coli. 3 to 6 subsamples were composited into a glass jar. And we did our in situ measurements at all the subsample locations for phycocyanin, chlorophyll, pH, specific conductance, those other physical measurements. And we took the samples right away, and we split and preserved them right on site in our field vans because we wanted to preserve them immediately, especially given the molecular methods. And so the molecular methods we use, this was kind of a focus of our study because we're a microbiology lab. So we did qPCR assays for cyanobacteria on 4 levels. So the first level is the most general, and that's general cyanobacteria, any type of cyanobacteria. Okay, so just a general population of your blue-greens. Then after that, we focused on assays for 3 genera of cyanobacteria that are widespread and known to produce microcystin, which is the most common toxin. And that's Microcystis, and this is a new name, Dolichospermum. Do we have any phycologists in here? Okay, that used to be Anabina, and that's the new name for it, and Planktothrix. So we looked for genes for those 3 genera, and number 2 was general genes, just for those 3 genera and either the non-toxic and the toxic strains. And then we went down another level, which is level 3, which is actually looking for a gene that is responsible for toxin production. It's called the MCYE gene, and it's genus specific for these 3 genera. And we looked for DNA for the presence of the toxin gene. And then the 4th level was the same assays, but instead of DNA, which tells you it's present, we look for RNA, which tells you have active expression of the gene. So 4 levels of cyanobacteria. Okay, so here are some of our results there were 3 lakes, 3 sites where we did the most intensive data collection, and we had the most data for data analysis. And they were Buckeye Lake, which is in central Ohio, Harsha Lake, and then a Lake Erie site, Maumee Bay State Park, Lake Erie. And these are 3 really different sites. And I'm going to show you right here. These are the toxin concentrations. This is the microcystin concentration, runs from 0 to 300. These are the number of samples. These are the percentage of detection. So we almost always found microcystin. And then we have 2 lines. We have this orange line, which is the Ohio EPA public health advisory of 6 micrograms per liter. And then we have the higher advisory level, which is a no contact advisory of 20. So to give you an idea, Buckeye Lake in central Ohio, it's a very shallow, it's an old canal lake, and it had the highest microcystin concentrations. It had a median of 55, which is really pretty high, and always above 20. And then in Harsha Lake, which is near Cincinnati, It's a deeper lake. It's a drinking water and a recreational reservoir. And we had a few samples above the 6 level, so much lower than Buckeye. And then this was our Lake Erie site. We had the widest range of microcystin concentrations, ranged from less than detection, which is 0.1, all the way up to 240. So when we started modeling, we decided, this is a little different than E. coli. So we had 2 different types of modeling scenarios. The first is the real-time models, okay? So those are variables that you have them right away. They're easily measured or continuously measured, and you can develop a model for real time, for right then, okay? But we didn't want to forget all the other factors that took a little more time, and we wound up, after going through several different names, came up with a name of comprehensive models. And so these are used factors along with your real-time factors. These are results. You have to take the sample, you have to bring it back to the laboratory. It takes more time, but it really gives you a lot more information about the cyanobacteria community structure, what's going on, and potentially what triggers toxin production. So 2 different types of models. And since this is the Great Lakes meeting, I'm going to show you the results from Maumee Bay State Park, which is in the western basin of Lake Erie. And we collected 24 samples that I'm going to show you that are in the results. Okay, so I won't show you too many statistics, but I will show you this. So we use Spearman's correlations as an initial data exploration tool. And Spearman's is a non-parametric measure of the association between 2 variables. So in this case, we're looking at the association between each of these factors and our dependent variable, which is the microcystin, because that's the most commonly found toxin. And we found some pretty strong correlations. So the row is the Spearman's results, and you want that to be as close to 1 for a positive correlation or -1 as possible. And these are pretty strong correlations. So we found for phycocyanin, turbidity, and pH, all these different factors, we found a row in this range, and it was significant, significance being less than 0.05. We also found a negative relation between stream flow in the nearby Maumee River and microcystin concentrations. So it looked pretty promising for our real-time factors. And then for the comprehensive factors or the comprehensive predictions, we found some pretty strong correlations there too. Microcystis, this is a microcystis-dominated system. This is the old method where you count cells under the microscope. We found a very strong correlation to microcystin. And then our DNA methods, the MCYE gene, and also general microcystis was strongly correlated. We also found a strong correlation between total phosphorus at the time of sampling and our microcystin, and some negative relations between some dissolved nutrients and microcystin concentrations. So pretty promising for predictions. But Spearman's doesn't tell you everything. You have to do plots of the data. And we looked at lots of plots. This one over here is the daily mean stream flow at the Maumee River in cubic feet per second. And those are these gray bars. And this is throughout the season. And then these red dots are the microcystin concentrations. And they go up to 240. So what this shows you is that during this period of low stream flow in the middle of the summer, that's when our microcystin concentrations were highest. We also do a lot of XY scatter plots, and I'm showing you a couple here. This shows total phosphorus versus microcystin, pretty strong relation. And this is our microcystis MCYE DNA versus microcystin, also pretty strong relationship between the two. So we saw these promising factors, and we only had 24 data points, and I certainly wouldn't advocate developing a predictive model from 24 data points. I know I've always said that. You need more data, but I couldn't resist. So I went ahead and so we used Virtual Beach. Y'all familiar with Virtual Beach? If you're not, it's a free software program provided by US EPA, available to everybody. It's great. It was written to develop models to predict E. coli. Okay. But you know what? You can use it for whatever you want. You could predict anything. So we, instead of E. coli, we put microcystin in as our dependent variable to predict it using Virtual Beach, and it worked great. These are the Maumee-based state park models that we came up with, and the R-square values, I realize this is 1 year, 24 data points, but it looked pretty good. We had some high R-square sensitivities in terms of the 6 microgram per liter threshold, and you could see the variables we used. So real-time comprehensive, then we did comprehensive models without the cell counts. So we've got the winners here were phycocyanin and stream flow. Wind speed at the Toledo Executive Airport came in, lake level change, and then our MCYE DNA came in on one of the models. But I want to use the remaining time to kind of highlight a difference between E. coli and our CHABs, and that's the use of continuous monitor data. These data were available from other sources. There was a continuous monitor suspended from a buoy at Harsha Lake, which is near Cincinnati. It's operated by US EPA, and it was about 1 mile north of the beach. Also, a Limnotech operated continuous monitor, and it was outside the Toledo water crib, I think about 6 miles east, and here's Maumee-based State Park. So we used these data from the monitors, and we had discrete microcystin concentrations that were associated with these data. 17 samples from Harsha, but only 8 samples from Maumee Bay, because this was a really small time period where they operated the monitor. But we went ahead and did it anyway, and looked at the Spearmans between, so what we did was we took, so it's continuous monitor data every 10 minutes, right? So we took an average of all those 10-minute measurements for the past 24 hours up to 10 a.m., which is the time of our microcystin sample, and we correlated it, and we found some really, really strong correlations. I mean, this one, 7-day phycocyanin, 0.98. I don't know about you guys, I've never seen that in the E. coli world, a 0.98 correlation. Some other ones that came out consistently were pH, pretty strong correlation. What didn't correlate very well was chlorophyll, and that's kind of what everybody's using. I mean, it did okay, but nowhere near what phycocyanin and pH did. So these continuous monitor data are really promising, and this shows that graphically it compares on-site handheld continuous measurements with continuous monitor. They're all significant, but they're different. And this is at Harsha, and this is at Maumee-Bay State Park. So in conclusion, the relations between all these factors were site-specific. That wasn't a surprise because it's the same way in the E. coli world. The continuous monitor measurements had the highest correlations, and we think it's going to work. We think it's really promising, but obviously you need to collect more data and on consecutive days. We only sampled weekly to get these relationships. I wanted to point out that we do have a report that should be out publicly available on the USGS pubs page like any day now. And these are our co-authors from Ohio USGS, Kansas USGS, and Pam Streffolino from the University of Toledo. We didn't do this ourselves. We had many partners that collected data, provided data for us, and here's a listing of them. It's funded by Ohio Water Development Authority and the USGS Cooperative Water Program. And I will leave you a foggy morning at Buckeye Lake. Thank you.
We've got time for one question for Donna.
Just Buckeye Lake always exceeded the levels. How many people would use it during this time?
Well, a lot of people
Do you have it
don't use
Could you
don't use the beaches because they're kind of green and kind of gross. But so what they do is they take their boats out, and there's a boater swim area, and they think it's clean out there. Well, we measured there, and it wasn't. So yeah, a lot of people do use, especially those offshore boater areas. Right.
We're doing great on time.
All right, great. And our final talk for the session is going to be presented by Frank Lupi, who's a professor of agriculture, food, and resource economics, as well as a professor of fisheries and wildlife at Michigan State University. Frank, thank you.
All right, thanks a lot. So you're probably wondering, what's the economist doing here? I was thinking that when I drove up through a little bit of snow in Cadillac today. But it does fit together, right? You know, economists are thought of as practitioners of the dismal science, right? And sometimes people argue with that some people don't think we're a science. So we don't get away from the dismal label. So then we fit with the talks by the CDC, because we're kind of a disease, we get that. Then I thought, well, we also are, I'm particularly interested in behavioral modeling, statistical models of how people use natural resources, so we fit with the modeling talk. And a couple of the talks mentioned sort of gaps in data. If you're an economist who likes to do population level modeling of how people use natural resources and you want to work on Great Lakes beaches, that's where the dismal thing is, because there's very little good data on people's use of beaches. So that's a good introduction as well, I think, to my talk, where we're going to try to attempt to place some values on beach closures that might be related to harmful algal blooms in Lake Erie. And this is a dissertation chapter soon, I hope, to be published at Michigan State University by Leah Palm-Forrester, who's now moved on to a position at Delaware. So she got out of the Great Lakes Basin. You guys know all the issues probably more and better than I do associated with HABs and excess nutrients in the Great Lakes. We're going to focus on the issue of beach closures, recognizing that there's many other potential economic impacts of HABs. In particular, we're going to do this in a data poor environment. We're going to try to estimate trips to Lake Erie beaches and the loss in economic value associated with when those beaches close, but we're going to do it in this data poor environment. There is a previous study specific to Lake Erie beaches that sort of meets economic standards for an economic model of beach use. It was published in the late 90s by Brent Sanjan and some folks at Ohio State University. It models a system of 15 beaches. We're not going to use that study. We're looking for something a little more current. So we're going to transfer estimates and models from other places in the literature and try to predict trips to Lake Erie beaches that way. So imagine you wanted to model water temperatures in a lake, but you don't have a model specific for that lake. You take a model developed somewhere else and you try to calibrate it to the individual lake. Understanding that this isn't a substitute for actually having the data, but we don't have the data, so we're trying to do something here. So what that is called in economics is benefit or model transfer. And we look at a couple of ways of doing that, and I'm not going to talk in much detail about that, but we're transferring estimates, but we're also transferring the models, and I'll focus on that. So what kinds of economic values are we trying to get at here? You know, there's different terms you might hear about economic values. The kind of values we're estimating are values that accrue to the people that go to the beaches. So they're not economic impacts to the businesses or gas stations or other types of effects. And we're specifically interested in what people would have been willing to pay to visit the beach above and beyond what they have to pay. So it's a net benefit idea that economists use. And what we're not doing is simply measuring the amount of money people spend when they go to the beach. So for an economist, the spending is a cost. It's a cost to the beachgoer. They would rather spend less and have more money in their pocket. We're trying to get at that net effect. So this is not a spending study. So back to what we try to do. There's two approaches to this idea of benefit transfer. We go to the literature, we try to transfer either estimates. So this first approach, we just take an estimate from somewhere else and we multiply it, say, by a number of users or number of trips to a beach. The second approach is this model transfer, where we're actually trying to estimate demand functions for all of the beaches, and that's what we try to do here. And the literature favors this approach. So where do we get these demand functions? From a study some students and I conducted in Michigan, where we collected data from the population of Michigan residents on Great Lake beach use, and we developed statistical models of where people go for Great Lakes beach trips. And those models are population level models. So they model both whether or not people take trips and then which beaches they go to on the Great Lakes. So it has this level of a model that's explained by people's demographics about whether or not they go to beaches, and a level of the model that explains which beaches they go to as a function of things like how much it costs them to get to the beaches, water temperatures at that beach at that time of year, and other factors. So these are temporal and spatially explicit demand systems. They're statistically complicated, but they're models. So that model that we're going to transfer was developed for 450 publicly accessible Great Lakes beaches in the lower peninsula of Michigan. The population of people in that model was Michigan residents. We're only doing day trips in that model. Why? Because, well, it was hard enough. So we know there's other kinds of trips. We know there's people from out of state that use the beaches. They're not in this model. It's not perfect. There's only 2 beaches in our model that are on Lake Erie. So it's really not well suited to kind of valuing the issue at hand for HABs. So we're going to transfer that model. We're going to take the relationships we built in Michigan and transfer them to the population of Ohio. So this is like a watershed, if you will. That's the market area for day trips to Lake Erie beaches if we allow people to drive up to 250 miles. So we're transferring the model to people that live in that sort of yellow area. And again, we're going to use it to predict single day trips and to value those trips. So on average, the typical person at an origin zone in this market area for the model has about 162 beaches they could get to in a day. So each origin zone in the model has 162 statistical demand functions. So there's a lot of these relationships in there. Where can people come from? We model that based on all the census tracts that are in this market area. So there's 4,740 census tracts. So there's 4,740 origins, all having about 160 demand functions. So it's a big probability system. And where do we get the data on those people? We get it from the census. So again, the probability of taking a trip in this model is a function of people's gender, their age, race, education, employment, income. It's omitted, should it be in that list, and children under 17. All of those things are available from the census ongoing every 5-year American Community Survey. So we're going to collect that data and use that to apply the statistical relationships developed in Michigan. So we're going to predict baseline day trips. Whoops. That's our first step. Then we're going to predict how trips would change if you close a beach. So in the economics world, when you close a beach, it's not like you might necessarily lose all those trips, Some people might decide to go to a different beach, a substitute beach. They'd take an alternative action. So behavior can respond by not taking trips or going to other sites. So we use the model to predict those changes, and then we use the demand relationships to get that willingness to pay that's lost when a beach is closed. So what do we find? A typical Lake Erie beach a day trip is worth around $16 to $18 in net economic benefits per trip. This is for day trips only, and these are the values that accrue in net to the beachgoers. A typical Western Lake Erie beach is worth around $2 million for the summer season. That's Memorial Day to Labor Day. In fact, that's the season for the model. We don't model trips outside of that. And again, these are the values to the people that use the beach. The users benefit when they have more access. We can make graphs. Here's a graph of the value of various Ohio beaches in Lake Erie. If we were to look at the lost value associated with closing one of those beaches individually, the beaches that are higher valued here roughly have more users. But what if we don't just look at closing one beach, we look at closing multiple beaches? So when you close multiple beaches, you take away people's opportunities to just go somewhere else. So that can have an effect on the estimated economic losses. And this graph shows that as you close more beaches at the same time, losses increase non-linearly because people lose close places they could go to instead. You're taking away behavioral responses or options that people would have had. The dotted line here is just a sort of this is kind of partly theoretical or methodological, this paper. That's what you'd get if you just did the simple multiplication. So transferring the functions in our study also mattered, consistent with the literature. So in summary, yes, we would actually rather have gotten data for all those beaches in Lake Erie and built a model just for Lake Erie. That would be much cooler. Absent the money or the data, we did this. So I just want to stress, this is model predictions. Transferring the model worked. It worked fairly well, it seemed. In our case, since only a handful of those beaches are, say, state parks where we have use estimates, it was essential to use the model transfer. Otherwise, we wouldn't have had estimates of beach use for most of those places. And then I made the point that when beaches are closing or are affected more generally over a larger scale, the losses increase non-linearly. So what's an important thing we didn't do? We don't have a variable to show how trips respond to different levels of HABs that are short of a closure. There's disamenities associated with HABs besides closing a beach, but we would need a study that had variation in HABs in the underlying data where the model was developed to get that relationship. That would be an important thing for future work. Leah, not me, got funding from Nature Conservancy and Great Lakes Protection Fund for part of her dissertation work. So we thank them. And that's all I have. Tried to keep it short.
We've got time for a few specific questions for Frank.
Yes.
Yeah, Frank, I've seen 3 other loss benefit transfer things, and the values are higher. They're like $25 to $30 for loss of beach day. Does the income level or the cost of living have anything to do with
the income level and the cost of living has something to do with it. Economists differ in the way they compute the cost of travel as well. And we use fairly conservative assumptions when I compute the cost of travel. We don't include the average cost of driving a vehicle, which includes annual depreciation and insurance and costs that just don't vary when you put miles on the car. We only use the part of that cost that puts miles on, or that the per mile driving cost, not the sort of total cost. When you take those things out, you get these lower values. So that's probably what's explaining most of that difference.
Anyone else? Oh.
Yeah.
Oh, Michael, do you have a question?
I'm sure it's melted.
Just a
Yeah.
quick one. What would happen if you built in the cost of prevented illness, diarrheal illness, from closing the beach, so people didn't go there and potentially get ill from something else?
You should repeat the question.
Yeah.
That makes sense.
So are you focused on if people don't go there, then they're better off because they won't get diarrhea?
Yeah.
So
the question is.
I'm tempted to make a pun about that
The
question.
question.
Well, there's
Frank,
a
could you repeat the
webinar.
question?
The question is, if you close a beach, some of the people that can't go there will benefit because they might not get a disease. It's possible. I don't know. I don't know what they're going to do instead. It could be something riskier. I don't know.
Thanks. So if all of the speakers could come up, and we're going to kind of have an open forum here. So like to get a little bit of a discussion going. So the one thing that I would ask is that as people ask questions from the audience, if one of the speakers wants to take a stab at the answer, if they would just repeat the question beforehand. Donna, Michael, Virginia. Yeah, standing is fine.
Yeah, we'll stand.
That's good. That's good.
All right.
So does anybody have a question for our group?
Leave this.
At the back.
This is actually for Donna. If you're designing a sampling plan where you're looking at those algal blooms, what level of intensity would you see as ideal given the dynamics and changeability?
You're just gonna have to come here and speak it.
Could you come up?
You gotta repeat the question to the webinar audience and then kind of speak.
Okay.
You can speak right in front of the microphone right there. You don't have to hold it.
Okay, the question was, if I was designing a sampling program, what level of intensity would I recommend? Every day is great, but who can afford it? So what we've typically done with E. coli, and what I think might work for the HABs, is try to get 3 to 4 consecutive days each week. So you can see what happens right before you might have a high toxin concentration. I mean, it would be great to do more, but realistically, that's what I would do. And I would do it, maybe sample less often in May and June. You might get away with 2 days a week. And then if things intensify in late July, 3 to 4 days a week, maybe even go 5 days a week, and then taper off.
Another question? Go ahead.
I'm curious if anybody's thought about ways to maybe crowdsource some of this data, self-reporting, or, when people go to the beach, if they see a bloom, or sick, those kind of things.
So the question is whether data could be crowdsourced about bloom activities at a beach.
You want to tackle that?
I'll go after you. Go ahead.
I'll say that Ohio EPA does have a system for citizens to report, either a dog death or an illness or green water or suspected harmful algal bloom.
Okay, so I think that I think there's a role for that. Would be my take on it, in terms of identifying, blooms that are occurring and communicating that up the chain. I think that how that fits in is something that I think probably needs to be thought of from kind of a surveillance data standpoint. In terms of there's still an evaluation component that goes into determining kind of what's a HAB and whether that's something that fits some criteria for surveillance. But I know that within some of the state health departments that we work with, they do look for information from their, from the public to identify blooms and illnesses. So I think that is a starting point right there. And the idea of citizen scientists is a really exciting idea for how to engage them in that work.
I was just going to add to what was said there that I think it's one of those things where you're not sure about the quality of the data, but depending on what you see from a social media standpoint and everything, can that lead to investigations then so that you can kind of focus your resources on where you want to go, especially if there's discussion on the social media side of different types of illnesses and that sort of thing. We're certainly interested in that, understanding that, I guess.
Yeah, just along those lines for Michael, is there another organism or pathogen you think we should be monitoring for in terms of beach health that we're not? Like, is E. coli or Enterococcus the right thing? Is there something that's more compelling?
So the question is whether there's another organism that would be more compelling or a better indicator that we're not monitoring for that we could be monitoring for.
Or not necessarily better, just something else.
Yeah, I mean, that's a $50 billion question or something like that. Is there something else? I mean, I think, again, we're focused on diarrheal illness, and so clearly that seems to be correlated depending on the method you use and that sort of thing. But, I'm always struck, and we were surprised by when we looked into the data of how many otitis externa, outer ear infections occur in the U.S. It's about 2.4 million per year. It's thought to mostly be waterborne and primarily Pseudomonas related when isolations are done. So, I mean, it's certainly something that could potentially be thought about. I'm not going to add any monitoring onto anything there, but I think it's good from a study standpoint to see how that's correlated, because we're seeing, about $500 million spent on it's easy to do surveillance for it because it's really, if you've had it, it's really painful. And so people show up at emergency rooms. And when we were doing this with the Academy of the Otolaryngologists, what I noticed in their magazine when they put our stuff out in it was all of the eardrops were for sale during the summer season. It's big business from a pharmaceutical standpoint. So it would be interesting to see whether there were correlations there. But I wouldn't talk about standard monitoring. Go ahead.
For Dr. Beach, is there a way to disinfect for Naegleria?
Oh.
Is there a way to disinfect for Naegleria?
Or prevent it, or like minimize it, or like we do for E. coli, we try to not get it in there.
Yeah. So is there a way to disinfect? You don't find it in pools if people run their pools appropriately. The only cases found in swimming pools are when it was really poorly maintained, Czech Republic back in the 60s. I think it's a tough thing to know how to control. This is a naturally occurring organism that's grazing in the biota in the lake. It's not that it's not human waste coming in. It's not these things. We don't see good correlations. There were studies done by EPA in the 70s and 80s. Not a lot of good correlates there. What I'd love to do, that we've been working on quantitative methods now to kind of quantitate it, is to go back through and kind of look from a population, meaning of lakes, to see where do you find it? It's thought to be ubiquitous, and they could find it at most lakes in Virginia, Georgia, Florida, and that. But during some of the big studies that USGS and EPA do and everything, you've got 1,000 other parameters that are measured. Could we measure Naegleria and try and do some analysis on those data? We would step up if you want to do something like that, because we don't have a good feel for the ecology of this organism. And I'd love to know a predictive model for when we could tell people there's probably a higher risk in this lake versus that lake, because we see it's in all the lakes, but you see infections here, you see repeat infections in certain areas, and it is not random where it's in the U.S. There are 3 tiers, kind of the California, the kind of geology of Oklahoma, Texas, and the Florida kind of southeast. They're quite clustered.
Do they have an idea that the amoeba lives in the sediment, or it likes to live at the top layer, or does it if the water's moving, it doesn't
right.
live there?
Amoeba in the sediment, does it move? Is it up near the surface? It clearly can be in the sediment because it overwinters. But because you find it in the same Minnesota lake, but you can see it in the water. You see it in the water column. A lot of what we see are infections. The average age is 12. It's usually young boys. We think there's a lot of a lot of action and activity and that sort of thing. You know, the birthday boy, where every kid at the birthday party shoved him under the river water. Shallow water, slow moving, seems to be better. You tend to see high peaks of temperature and low flow, and the river infections tend to be there. But there's a lot of ecology that we don't understand because it's kind of an unfunded mandate. But when this hits a family, this is, and it's for those tragic the individuals who tragically lose a child, but it's also for the, especially Florida and Texas. This is not great when you tell people in Florida your lakes have Naegleria, you know, you could die. So there's a much more there's much more of a societal issue here where it overwhelms the health department and it really impacts tourism when you have a death. And so it's a tough. Give us your ideas. What do you think we can do here? It's a tough one.
Yes.
Just out of interest, does Naegleria affect animals?
Does Naegleria affect animals? Yes. So we've seen it in multiple mammalian species. Cattle trough, warm water, cattle all dipping their noses in. South American tapir died in a zoo from Naegleria. And the model is actually the mouse. You know, mice don't swim very well, so you put Naegleria in the water, put the mouse in, and it dunks itself under, so it inoculates. So it clearly affects other mammals. There's other Naegleria species, but only Fowleri is known to cause human disease. Yeah, Richard?
Let's see, I have a question for the economist again. One question is, when's the dissertation going to get published? From the student that went to Delaware, is it?
Yeah. So the question is, when is that dissertation going to be published? She has a job as a professor there, assistant professor, so she's got to turn it in shortly, like before December 14th, certainly. But we also have a paper under review. Hopefully it gets accepted, and then we'll share it. I should also, given the chance to talk in the microphone, qualify my answer to your earlier question. Had we built into the model these longer distance trips, we would also see higher values per day. Those lower values per day are only for the shorter distance day trips. Just context.
Another question is, let's say the city of Houston or Miami wants to replicate your study. What would you tell them the net cost, including overhead and so forth, would be for a study?
The study that was done in Michigan, we cobbled together probably $800,000 to collect that data. There were 30,000 surveys sent out. It was an extensive effort put together with money from related grants that I got from EPA, NOAA, coastal ocean programs, everywhere we could find bits and pieces of funding. But the survey, just the survey effort itself was extensive. The model development for that model is extensive. It takes a long time to run. It's not trivial. So it was a big effort, and we're still trying to work on making it better. If you do a study at one beach, it's very different. We're trying to characterize beach use for the population. So we're trying to capture every, all the places people could go.
My question's for Michael. What's the methods for detection of that amoeba, and is it real costly?
It's a PCR-based, molecular-based method. So diagnostically, we have all three amoebas in one test sort of thing. Environmentally, it's an immunomagnetic kind of capture and concentration. So large volume concentration, immunomagnetic beads being used, and then PCR for that method.
It's not something that water or plants or anything like that would ever probably even test for, is it?
Yeah, and the question was about methods and what methods there are. Yeah, this is not something where we would recommend routine testing. In fact, we actually discourage it because we think that people will find it versus where, it's like, so, I mean, I think, and I know it sounds kind of funny, but what do you do? It grows naturally in all these lakes. So if you find, you're going to close every lake that you find it in, or you're going to put a sign up, or you're going to put a sign every 10 feet. You know, the data says from day to day, hour to hour, it can differ whether you find it. And so really we focus in talking with state and locals is it's where the infections occur, because there's something else we don't understand there, and that, you know, you want to think about what you do with signage there. But to put a sign on every lake that has Naegleria means that the state of Florida will have a sign on every single lake, and where on the lake should it be? So it's again, as a naturally occurring organism, you know, we don't go to the ocean and say there be vibrios here, or there be sharks in here. You know, we've got to, you know, you got to be a little bit more realistic, I think, long term. So.
Michael, can you explain, though, some of the health departments, they just say, plug your nose. You just, like, there's a cultural educational message in that.
Yeah, I mean, again, this is a relatively rare infection. Therefore, we will never have good data to say what really prevents this. Makes sense if you plug your nose, and if you're going to jump from the top of the slide or something like that, to not have water jetting up there. But we don't really have data to say that you couldn't slide into the water and have the amoebas go up. And so, you know, you have to take, what we say is, think of it, it's risk communication. The unknown risks are the scariest. Your highest risk was driving to the beach. But if you're the one who gets it, you're not in a good situation there. But it's a relatively low risk compared to many of the other risks we took to go there. You're much more likely to drown or to die in a traffic accident on the way to the beach. If that makes sense.
All right. Before we finish up here, I just want to remind everybody there's a break coming up down by the vendors and exhibits. There's some snacks things, so if anybody needs to refresh, there's going to be some refreshments down there. Before you leave, though, I'd like to give another round of applause for our speakers today. And the next session starts at 3:30.
Yes, let me turn this off. Oh, you're here already.
I get it.
Yes. Yeah, freaky.